Hello everyone, my name is Dr. Graham Cottrell. I'm an Associate Professor in Biochemistry, and welcome to the first of 3 videos on the molecules of life, in which we'll focus on proteins. So the purpose of this short video is to introduce you to the molecules of life, and one of these is important one are proteins. So we're going to discuss the diversity of proteins, both in terms of their shape and structure, but also their function. And we're going to discuss the types of amino acids. These are the molecules that go together to build up a protein. So the different types of amino acids that are found within proteins. So what are proteins used for? Well, proteins are used throughout the whole body. You can find proteins in your blood, and here an example is hemoglobin. Also in your brain and nerves. These are small signaling molecules. Enzymes, which are biological catalysts, and they speed up the chemical reactions that occur in your body. Antibodies are proteins, and these are made of amino acids, and these can help your body fight infections such as bacteria and viruses. Muscles, everyone knows muscles contain protein, and it's the interaction of actin and myosin which enable you to move. And here are some examples that we'll probably come across in the rest of the course. So an enzyme, an important enzyme in the body, is called hexokinase, and this is involved in the metabolism of glucose, which you'll learn about later in the course. Transport and storage. Hemoglobin carries oxygen around your body, and you'll cover this in the lectures relating to blood. Coordinated motion. So without the proteins actin and myosin in your muscles, you wouldn't be able to move. This also provides mechanical support, and here the protein collagen plays a big role. I mentioned before, immune protection, so antibodies or immunoglobulins are critical for helping you fight infections. Antibodies are also used in diagnostic kits and in the clinic for testing for particular diseases. Generation and transmission of nerve impulses. So receptors in the body that respond to signals are also made of protein. And they also control the growth and differentiation. So many of the factors that are produced by your body that stimulate your cells to proliferate or to grow and divide are made of protein. Proteins come in all different shapes and sizes, and here in the left panel we can see collagen. So collagen is like a 3-stranded helix. Each separate strand of the protein is wrapped around each other, and this gives it its strong structure. Antibodies are Y-shaped proteins, and the portion at the top, here coloured yellow, this is the part that recognises a specific antigen or another molecule. And these antibodies will send signals to other parts of your immune system that help recognize and then destroy invading pathogens. DNA polymerase, so you should have come across this in Dr. Madera's lectures. And here we can see a donut-shaped protein, and you can see the hole in the middle, and this is where the DNA will pass through as the DNA is replicated. So you can see that there's very, very different structures depending on the role of the protein in the body. So as well as very different structures, they all come in different sizes. So the largest human protein is called titin, and this has a massive 34,000 amino acids. Smallest proteins are a little bit more controversial, but these are usually considered to have less than 100 amino acids. And it's controversial because usually quite often they are processed. So they are made as a longer polypeptide chain, so a long chain of amino acids, and then they are cleaved by proteases into these smaller proteins. The very smallest one that we know of is glutathione, and that has only 3 amino acids. And there are other very small proteins, often called polypeptides or just peptides. And these are the enkephalins, and these are important in regulating nociception or the sensation of pain. And these bind and act to opioid receptors. So what are proteins made of? Well, proteins are made of molecules called amino acids. And here in the diagram we can see a generic amino acid. On the left here we can see the amino group, that's the NH2. And on the right there is a carboxylic acid, hence the name amino acid. The R group here is unique to each amino acid, and this can change in its size or length, and the chemical characteristics of this R group give each amino acid its unique function. So there are many hundreds, possibly thousands of amino acids, but in eukaryotes or in nature, then we only use 20 different amino acids, and all of these are of the L stereoform. So we class these chemically according to the type of R group, so whether it's hydrophobic or hydrophilic, but they can also be classed based on how the body synthesizes them and the diet. So these can be termed non-essential, and these are amino acids that the body can produce, whereas essential amino acids must be taken in from the diet. There are also some called semi-essential amino acids, and these can be produced by the body when necessary, but they are not normally, and usually they will be taken in through the diet. Often in the literature, the amino acids will be written in shorthand, and these are given a single letter or a 3-letter code. Now, it's not required that you memorize all of these single letters or the 3-letter codes, but it will help if you're familiar with some of them. So we do have charged amino acids. So these are often used within proteins to make salt bridges. And you can see on the left we have an amino acid called lysine, and the 3-letter code for this is LYS, and the single letter code is K. And this has a amino group as part of its R moiety. On the right we have glutamic acid, again because this has a carboxyl group here, and this is symbolized by the letters GLU or E in the single letter. So they can have positive or negative charges, and you can understand that these charges can then be used in chemical reactions, perhaps within the active site of an enzyme, or even bonding together, as I said before, and these can make salt bridges. Then we have polar residues or polar amino acids, and these can participate in forming hydrogen bonds. One of these is called serine, and it has an alcohol group and an OH group, and the short letter is S, or the 3-letter code is SER. And we also have cysteine. Now cysteine is a quite important amino acid because it's often used in the formation of what we call disulfide bonds. So this is a sulfur-sulfur bond, and this is quite often used to attach 2 polypeptide chains together. Hydrophobic residues, so these will usually have methyl groups on the end, so these don't like to be exposed to water, and these are quite often buried deep inside the protein when it's folded. Example here is alanine, or ALA, or A for short, and then methionine, also known as MET or M. So methionine is the amino acid that usually starts off the polypeptide chain for every protein that's made. So it's usually the first amino acid of every single protein. Then we come to an unusual amino acid, which is actually not an amino acid, it's what we call an amino acid. So you can see from the structure of this amino acid called proline, or pro or P, that the R group actually is attached back to itself. So this gives it a cyclic structure, and this can often influence the way that a protein folds. So that when the polypeptide chain kinks or bends, there's often a proline there, and this allows the polypeptide chain to fold back on itself. So this contains a secondary rather than a primary amino group, and hence it's termed an amino acid. If you'd like to learn more about the amino acids that we've talked about in this video, there's a very good chapter in Molecular Biology of the Cell, Albert's chapter 3, and also in Biochemistry, Stryer in chapter 2. Thank you all for listening.